Very much, Prof Suresh, for your time to discuss our project in the 2023 Indonesian iGEM League. First of all, the most important question of all. So, for those who don't know, I am a student under his supervision. So, we want to know what he thinks of me. So, am I a good student in your lab?
Oh, you're an excellent student, Michael.
Oh, thank you. Next question.
But not all the time, but that's okay. You can always improve on.
Okay. Thank you for the honest feedback. Okay.
You know what? I always say, like what the lab would like to have, the motto of, the motto of Rolls Royce. Apparently,
Rolls Royce.
Someone told me this. I'm not sure. Someone said they had a motto of we want give us the best, we get the best, and we make them better.
Yeah.
So I would like to believe that my lab is something where the best come and get better.
Nice. So for those who wants to join Prof Suresh's lab, you make sure that you're the best. Okay. Anyway, so can you tell us more about your research? I mean, I already know what your research is about, but can you give us a brief walkthrough to your research?
Well, I think I'll just make it a bit more simpler. So we have primarily three areas of research that we focus currently in the lab. So they're all somehow related to temperature to begin with, to start from there. So one of the key questions that my lab is interested in is we try to understand how plants sense and respond to changes in temperature. And when we talk about temperature, we're not necessarily talking about large changes in temperature. We're talking about small changes, maybe a couple of degrees here and there. So that's one of the key areas. We would like to understand what are the genes that control this process, what are the regulatory mechanisms, etc. Because we don't really understand everything about how plants sense. For instance, when in spring, we know that plants flower. So this is related to both increase in temperature and increase in day length. So we understand a little bit about day length, but we don't necessarily understand the temperature parts of it, how it is sensed, how it is transmitted, and so on. So we study that process. We study this using classical mutants. We study this using natural variation, which is basically the same plants from different parts of the world experiencing different temperatures, how they are reacted if you actually bring them to your same temperature condition, for instance. And a lot of our work is actually underpinned by considerable work on computational biology and genomics. So that's the kind of a starting point for us. But then when the system that we study is a plant called Arabidopsis, which is, you can consider that like the fruit fly of the plant world, if you like. Fruit flies are the model systems for the animal world in some sense. So the good thing is there's lots of resources in Arabidopsis that's grown in different parts of the world, so we can bring them to the lab, give them identical condition, we can see what kind of changes exist. Interestingly, when we actually tried to do this type of analysis, we found some accessions or some strains, as we call it, from Ireland show a very unusual phenotype. So if you grow them at 23 degrees, we have several strains where if you grow them at low temperature, at 23, they show a particular type of phenotype. But if you grow them at slightly higher temperatures, 27 for example, sometimes they don't grow and sometimes they show very unusual phenotypes and so on. So we study this process. And one such example led us to discover the first and trinucleotide repeat expansion that is associated with a growth defect. And we know triplet repeat expansions underlie many human genetic diseases. The specific type of expansion that we looked into plants turned out to be something related to this expansion that is seen in a neurogenetic disease known as Friedreich's ataxia. And there is no cure for the disease, there is no treatment at this point. So we thought we can utilize some of the plant system to try and understand some molecular mechanisms, which we can then explore in the animal systems. So we studied that in the plants, and now one of my former lab members has their own group studying this. But my own group actually now studies how Friedreich's ataxia is actually how Friedreich's ataxia is controlled in the context of using mammalian cell culture, for instance, the mechanisms and so on and so forth. So that's the second part of the work. Third part that sort of grown up from here is like when you grow plants at high temperature, one of the things that we saw was there are changes in the molecular process known as splicing. And then when that sort of got us into the field of splicing, and then we started to look at it in a computational way. And so there is a considerable proportion of my group currently, including Michael himself, is actually in this space of genomics and computational biology of splicing. That has got wide-ranging implications from agriculture to medicine and from plants to humans. So we study this using Arabidopsis, Drosophila, and human systems, and this involves a lot of natural variation studies and Jiva studies in human system, in flies and in Arabidopsis, and we are testing many of these experimentally, and there are some very fundamental exciting work that's coming out of that work. So that gives you an overview of the different things that my lab is currently focused on.
All right. So it stems from the curiosity of how temperature shapes plant development and growth, and also reproduction, and then it branches to multiple different areas of research.
One of the coolest things of being a geneticist is you are just curious. You just follow what you can actually study, and if you find a cool phenotype, cool phenotype in a classical sense is a messed up plant, but if you want to try and understand what happens there, genetics, the science takes you to your next step. You don't necessarily think about it, and just curiosity driven fundamental biology which we believe many of which will have very wide ranging implications to understanding how life works.
So our project specifically focuses on the role of carbon emission as one of the leading causes of global warming, and how to take that into resource that can repel insects. So we're trying to engineer a type of yeast called Pichia pastoris, and we will turn them into autotrophic machines that can stimulate carbon dioxide and then turn them into an naphthalidone, which is a carbon-based compound that can repel insects, including mosquitoes, that are responsible for mosquito-borne diseases such as malaria. Like you mentioned earlier, that even if with small increase in temperature, plants will exhibit large difference in their phenotype, and especially when we are talking about global warming, the increase is not significant. To be fair, it only increases about 1 degree per century probably. So
I think the thing to recognize, when you talk about the impact and the temperature impact or any kind of impact for any environmental perturbation, that's a matter of sensitivity. So, for example, to give you an idea, a real life example from my own work, I'll just tell you something. So, light is sensed by a receptor called photoreceptors, alright? So, we discovered that there is a difference. There are two different versions of a photoreceptor called phytochrome C several years ago on our work. And if you look at the distribution of these two different varieties of the receptors, they showed a different kind of latitudinal climb. It turns out that where there is less day length changes, so you can have in the part of the world that can be day length changes can be less, and there is part of the world the day length changes can be very high. And depending on where you are, the light receptor distributions also differ. So sometimes, in order for you to sense small changes, you just need things to be more sensitive, therefore they can be sensed. So it's very kind of difficult to generalize. It's a very there is a significant impact of something looks like only 10 degrees is significant. That may be our perception, but in some cases 1 degree makes a difference. For instance, the plant yield reduces substantially. Every 1 degree increase leads to a substantial reduction in yield, several percentages. So it differs from crop to crop, but a single degree increase in temperature actually means a considerable reduction and the reduction in the crop yield can actually translate to considerable number of people's lives. So the impact is something that you need to think a little bit more carefully. It's one thing to say a statistical impact of temperature, blah blah, but it's a completely different thing to think about the actual impact it might have. So you got to be a bit more careful, I would say. But that's fine. It's okay.
Yeah. So about this, about the findings you got from the temperature studies in Arabidopsis.
Yeah.
From the results, what can you extrapolate to the real world, especially with overpopulation and?
I would say Arabidopsis is the real world because it's not some artificial world.
No, I mean, in terms of other.
Arabidopsis is okay. So we're studying a plant, it's a small plant, but it is still a real plant. It is not some.
It's like a simulation.
It's not a simulated plant, and it's not like that. So it is real world in that sense. But I think, jokes apart, I think I will take that question as like you're asking, okay, you're studying a model system, you're looking into the small changes in temperature, what does it probably mean to crops, for instance, or crop yield or something like that. I think the way we need to understand this, or at least the way I think about this is this. So Arabidopsis gives us a handle. So when we study any model system for that matter, we use that because we can actually get to those genes quickly. So I would say that many genes that we are currently discovering in crops or any other plant species for that matter, it would have taken us even much more longer than what it is currently taking, because we have so much information accumulated in Arabidopsis system. So it overall speeds up the process of understanding the real world. So that's the biggest impact in the first instance. Specifically coming to temperature, for instance, the first thermosensor, if you like, we discovered, I wouldn't say we, like the plant biologists, my colleagues, Phil Vege's lab, for instance, together with Jorge Casal's lab in Argentina, they independently shown that phytochromes are potentially temperature sensors. Phytochromes are known to be light receptors, but they also showed that that one of the chemical reactions involved in phytochromes is sensitive to temperature, and therefore that allows them to sense the temperature as well. So we have other temperature sensing mechanisms that's been proposed, RNA structure being one of them. So there are a few others examples that exist. So those genes and those thermosensors do exist in other systems too. There is a role for epigenetic gene regulation that exists in other systems too. So studying Arabidopsis gives you the handle, and it seems many of the fundamental processes are often conserved enough. Of course, there are species-specific differences, so it is not like there is a reason why a rose is a rose and Arabidopsis is an Arabidopsis, but these fundamental mechanisms that one is uncovering in any model system obviously has a direct impact. So that's why, for instance, we are studying repeat expansions in plants, and the epigenetic mechanisms we study we believe have an impact in the context of human diseases. So that is going all the way from plants to humans. In many fundamental principles that we are uncovering in our study, which is yet to be published in the splicing, seems to have very strong connections, even as from between kingdoms, for instance. So obviously it has very strong impacts on the real world. I think that's why it's important to recognize it in that context. Of course, we also recognize the limitations of it, because obviously, as I said, rose is a rose and Arabidopsis is Arabidopsis.
How about the underlying molecular mechanism that is responsible for different phenotype in different, like in increasing temperature? What's going on in these plants?
Okay, so there are two mechanisms that I'll try to explain to you. One of them is came from a study of one of my very first students, who runs a lab right now in China as a professor. He's a professor there, and Wang Sheng is his name. And Wang Sheng, when he came in, he discovered that one of the plants that when we grew, their first leaves were just not getting formed. They were fine, they were germinating fine, but the first leaves won't be formed, and if you let them grow, they'll be very, very small. And when we actually cloned the underlying gene, we interestingly call that genus Icarus. Are you guys familiar with the story of Icarus?
The Greek person who wants to fly
close to the thing, so basically too close to the sun, and so it's sensitive to temperature. So this Icarus, we call this mutant as the Icarus mutant. So this is a natural plant, and in this case it turns out that basically when the temperatures increase, what the plant does is, instead of, so normally, you know, in the growth you have cell division and then differentiation and keep going on, right? But in this plant, when you have mutations in the Icarus, what happens is that you instead of going through cell division, you replicate your DNA, but you don't go through cell division. So you end up forming an endoreduplication mechanism. This process is known as endoreduplication. So a majority of the cells are just the DNA is getting replicated, but it just stays as it is. So it's almost like an avoidance mechanism. So when the temperature is very much high, the plants just don't grow. And then when you actually get the temperature back normal again, then these remaining diploid cells actually go through the division and actually start growing again. So here is a mechanism of a sort of temperature avoidance mechanism. So if you go into the natural conditions of where these kind of plants exist, they exist in hot climates in Spain, for instance, these specific alleles. So it is a kind of a evolutionary temperature avoidance mechanism for the plant to sort of, if it's too hot, I'm just going to keep quiet, and then when the plant condition is fine, I'll go back. So this is one mechanism, for instance. The other mechanism which we looked at with respect to repeats, for instance, is like we could show like what happens when you have the repeat expansions. You have the genes getting silenced, and then in this case, the mechanism that we found out was that when you have high temperatures, these repeats somehow lead to increase in small RNAs, and these small RNAs then target that loci through a pathway known as RNA-dependent DNA methylation pathway, bring in all the other components and actually silence this in the locus. So here is two examples why you are seeing the phenotypes that you are actually observing. And then of course there are other phenotypes, like for instance, very well-known phenotypes. Like one of the phenotypes that we study is when you grow plants at low temperature, you have a shorter hypocortical. When you grow plants high temperature, you have a longer hypocortical. And that we know, for instance, is when you increase the temperature, that leads to expression of some of the transcription factors, which is required for the synthesis or increase the transcription of the genes that encode for synthesizing the plant hormone auxin, and then you have an increase in the auxin levels, and once you have an increase in the auxin levels, they lead to an elongation. So essentially the mechanisms are reasonably worked out in many of these instances, but some of the phenotypes that we see are the phenotypes are new, and therefore it allows us to identify new genes, new pathways, and new mechanisms. And we obviously believe many of these are very conserved across the species.
Really interesting. For those who might remember, I think I've told Prof Suresh about this as well. That paper about the trinucleotide repeat expansion, I actually translated it into a worksheet for the Indonesian National Biology Olympiad selection. Felda was one of the students who actually worked on that assignment. Yeah.
Congratulations on that.
Thank you.
You remember that, Felda?
Yeah, it was a killer.
it was crazy. Yeah. I think the highest score is like 60 or something like that. But anyways, that's the kind of interesting research, and especially the target market for our organization originally is our Indonesian high school biology Olympiad students who might be interested in research, and because the Indonesian government has just launched a new scholarship scheme for international for these students to undertake international studies. So we hope that if they see these videos and they're interested in your research, they can these great talents can come to your life and like, hey.
Of course. I think we always look out for the best people. Actually, to be honest, the word "best" is something I use that in the very beginning as a punchline, but. I'm looking for motivated, enthusiastic students. I think motivation is the most important thing, and as long as the students are interested and motivated, I think they will be able to. It's just about having the right environment for the flowers to flourish.
So these students will be motivated because they've got motivation to make them study all about biology. So I'm pretty sure they're interested in your research as well. But so now we transition to our project. So the technical aspect of our project. So basically, in the Pichipastres yeast genome that we are going to use, we will introduce certain genes that make them autotrophic. It's the XUMP carbon cycle, and then also inserting the genes responsible for the conversion of that glucose into the naphthalactone inside propellant compound. So we've made the genetic circuit here for you if you just don't mind to just look at it. So I know there are so many genes involved. Well, fortunately, this is a design competition and not a hands-on competition, because otherwise we will be doomed. And then these are basically the CAB cycle genes. So, I think a research group in Germany actually found, actually made this discovery about turning this yeast into an autotrophic compound. And then basically this is the biosynthesis of naphthalactone genetic circuit, and also. So most of them we just use the original promoter, but for this one we use a different set of promoter because we're trying to match the rate of glucose synthesis with the naphthalactone biosynthesis as well.
Do you know whether if you're using different promoters, do you know whether those promoters are actually regulated through feedback regulatory loops?
We have no idea, honestly, but
that's something you might want to check.
Okay.
Because, for instance, you're using a promoter that's actually under auto-regulation. So the moment that something is increasing, then it goes and shuts off the thing.
Yes.
So if you have something like that, I'm actually not sure whether I'm supposed to tell you all this. I don't know.
Yeah. That's what
I don't know what your iGEM competition rules are. So
no, it's
I'm
fine,
not sure, but basically. That's something I'll think, if you are going to use some promoters, and if it is under auto feedback regulatory loop, then it will obviously shut down. So if you're trying to increase it using it you got to be careful.
Something else that we take into consideration is that these promoters are basically constitutively expressed promoters. So we just take the list and say, these are constitutively expressed, and using these toolbox we try to assemble these. And then also this is the kill switch.
If you have using constitutively expressed promoter, why there are so many different promoters?
Well, for to be honest, this one
I shouldn't be actually asking you the questions. I suppose. I'll leave it.
The CBV cycle, we just take the circuit
That's
from
fine.
the paper.
You don't need to answer me. It's fine. I'm just giving you.
But I think it's something to think about as well.
Okay.
I think it's because we want to.
Maybe it's all this. You have all the reasons there, but you don't need to tell me.
And anyways, this is the kill switch pathway, which includes the toxin relay antitoxin system. So we're going to supply continuous amount of tetracycline in our bioreactor.
Yeah.
And then within that condition in the bioreactor, it will produce antitoxin to repress the activity of the toxin. But then once, let's say there's a spillover and the microorganism actually went out of the bioreactor, then the antitoxin producing system is not there, and then the toxin will slowly kill the cells. So it minimizes the potential impact of this organism in case of spillover.
Okay.
So do you have any other insights or feedback on this?
Well, I think I just gave you as you were telling me a few different suggestions. I think that's
okay.
Yeah.
And I think that's all from the genetic circuit part. And so basically, what we are trying to integrate this into the genome of the Pichipastres itself. Do you have any?
Well,
idea?
I mean, what I understand is you already have somebody who has done this.
Yeah, somebody has done this. I think they used Cas9.
So the question is if it's already done, unless there is a specific reason to change, or if there's some efficiency issues that you can improve on, I don't think this is a problem, right, per se.
Do you think there's other there are other mechanisms that is actually more effective than Cas9 recombinant?
Look, I don't know specifically about Pistia pastoris, so it's kind of hard to say it from there. Because I'm not an expert on that. So I think I should stop giving you advice on things which I actually don't know much about. So it's actually a better thing to do.
Or probably.
But I mean, if I were in your position, probably I will be looking at what people have done and see if there is any specific bottlenecks are there. At that point of time, one can look at, okay, look, there is an issue here. Is there a way that we can improve this specific aspect? But if there is no bottleneck, if it is a standard process that I know others have done it, then the only question is if others can do it, we can do it too.
Yeah,
so I'll just go with that. Yeah.
Okay. Another question. Okay, you can see that these all these insertions are crazy, and it would be actually really hard to actually do it if we are actually.
I think the people are in terms of genetic engineering, there's a lot more things going on. So in the context of, in this case, you're actually trying to put a few pathways in there. So in the big scheme of things, yes, it is a little bit complicated, but it is not something that's like, very tricky. So
would you think this is actually feasible? Like, this is actually achievable?
Well, that's a very different question, because I actually, if you're asking me whether to put these genes into Pistia pastoris is easier, yes. But whether it would actually do what you want it to do, that's a very different question, because the functionality of each of this requires specific testing.
Yeah, so
and whether these enzymes are sufficient, whether, for example, one of the things that you're assuming is like the organism by itself is not going to do anything about you putting in there. But we don't know that, right? Yeah. So, you're putting in something else, and, what impact it would have on the fitness of the organisms and all that, we don't know. So it requires obviously testing. So theoretically, yeah, perhaps it might work, but practically, you can only talk about whether it is doable. If somebody else, if you ask me, I'll say if someone else is able to put some pathways into the Pistia pastoris, you should be able to do that too. But whether that pathway will actually operate in the same way, and whether it, your enzyme, one of the things to test is like whether your system actually has got all the components required for it completely taken up. Because maybe something else is required from the host, and if that is not there in Pistia, then it won't work, right? So you need to look at all those kind of things. But otherwise, I think it's definitely worth trying.
Okay.
Yeah. I'm sorry that if I'm not giving you.
That's definitely alright. Well, it actually makes sense. Like, I think this is one of the beauty of Indonesian League is that you don't have to actually perform the experiments. So there's no limit to your imagination. Like you can imagine as well as you want.
Yeah.
And we're planning to perform mathematical modeling to simulate the effect of these new enzymes and genes, and also to perform metabolic simulations as well. So, do you have any safety concerns regarding these microorganisms in your opinion?
No, I guess it is a standard, safety level containing, laboratory based work. So I think, per se there isn't anything that I can immediately think of. There's none of this looks like a pathogenic component in it, as far as I can understand.
Okay, so
that should be a standard genetic engineering practice. What I kind of not track, like standard genetic engineering project.
So you'd probably say that this project can be performed in like
BSL1, PC2, or
PC1,
maybe
PC2.
something like that, PC1, PC2 types of things.
Yeah.
In the Australian context.
Yeah. All right. And we're talking about the large scale of the grand scheme of things in application to worldwide scenario.
Mm-hmm.
Like you yourself interested in the genetics of plants.
Yeah. What I wouldn't say genetics of plants. I'm interested in genetics.
Yeah, genetics. But okay, genetics. But we'll talk specifically about plants. And like these kind of organisms.
Yeah.
Can you think of any negative effect of these plant-based GMOs, or microorganism that has been altered genetically?
Well, I mean, when you alter any genetic organism, I think you just need, there are control mechanisms in place, regulatory mechanisms in place in terms of actually analyzing it in the context of public use. So as long as we are actually fulfilling those safety requirements, I think we should be okay. But the specific modifications and the specific changes and the specific advantage that might actually provide to the organism over, and how it might actually affect its success in a particular environment, all those things require some testing. So as long as the testings are carried out, it's proper, and you're able to, there are different committees that will actually look into the specific aspects, we should be okay. I don't see anything exceptionally different and difficult in this particular project compared to any standard GMO-based approaches.
All right. So, do you think your lab is probably interested in these kind of research as well in the future? Like,
yes and no. I mean, we have our own ways of designing things, which we wanted to change. It's not necessarily this specific type of an approach, but we obviously are interested in generating climate-proofing solutions, for instance, right? The crops that actually can withstand and grow in multiple diverse different temperature conditions, and there are many different ways of doing that, and GMO is one of them. It's not the only solution, but that is one of them, and we need this, so there are a lot of design genetic engineering based solutions for sure we are interested in, and we might be wanting to, for instance, there are ways in which we can actually alter splicing, some of them genetically, some of them non-genetically, and that actually has direct implications in the context of disease response in plants to genetic diseases in humans, to all kinds of things, agriculture to medicine. You have wide-ranging implications, and therefore in that context, yes, definitely my lab is interested in this kind of work. But I would still consider my group to be a very fundamental biology group than
an application,
than an applied based thing. So of course we are not blind to the fact that some of our work actually have applied solutions to it, and if there is a appropriate opportunity, we will definitely take it to that mode. But a lot of it is we are probably the ones who are I think we are the basement builders and not necessarily the builders.
So you're basically creating the blueprint instead of building the...
I think that's how I would probably consider our group.
Okay.
Yeah.
All right. I think that's all from our side. Thank you very much for Prof Suresh for providing some time for us to interview you, and we